Okay, so hello, we are Group 5, and my name is Justin Ritter. My partners are Alexander Williams and Parker Edwards, and you'll see them later on in the video. But here, I'd like to introduce our book. So the book we did our video project on is Neanderthal Man, in Search of Lost Genomes, and the author of this book is Svante Pääbo. Okay, so now we can look at some background and context of the book. Pääbo writes this book from a first-person point of view. So he is telling his story to us and in the first person. He is speaking about his career as a gene scientist and specifically of his search to complete lost genomes. So and the lost genomes he was researching was mummies, dinosaurs, and Neanderthals. So he was looking to come up with a complete DNA sequence of these topics. That's what a genome is. And specifically, the Neanderthal is what our video topic is going to discuss. We'll go into how he came up with the genome and how it relates to our genome as Homo sapiens today. So our topic for our video project is how gene scientists have extracted Neanderthal DNA and how it relates to us as humans today. So we will talk a little bit about some of the research techniques used to extract Neanderthal DNA, some of the critiques about that research, and then we will compare and contrast the genomes, complete genomes of Neanderthals and humans.
So now we're going to talk about how these gene scientists are able to extract the Neanderthal DNA. So this is done through silica absorption. So this is an extraction method which uses the Neanderthal bone powder and is left in a buffer solution overnight. After this period of time, the Neanderthal DNA will separate into the buffer solution from the bone and collagen. Then this DNA can be run through silica surfaces, which we are able to bind the DNA molecules to. And this is how gene scientists are able to extract the Neanderthal DNA from their bones. So now that scientists have separated this Neanderthal DNA, how they completed a full genome based off this separate DNA they've constructed. So they have done this through a method called the Sanger method, which determines the nucleotide sequence via a DNA polymerase. So the DNA polymerase is just an enzyme that uses the old DNA as a template and creates new strands of DNA. So now that the gene scientists have completed the structure of the Neanderthal genome, they wanted to now see the difference and compare the Neanderthal genome to the human genome. So when comparing to these two genomes, they see that there's a 997% identical percentage between the two. However, the 0.3 difference leads to genetical differences such as cognitive development between them. There is a different skull structure between our skull and the Neanderthal skull, and also the energy metabolism used between the two.
So for current research today, there's not a whole lot going on since the major breakthrough was actually sequencing the Neanderthal genome. But one of the main things we can look at now is actually how they went extinct. So for our first thing, we have climate change, which basically led to getting an ice age, which was colder. Animals would die off. It would lead to starvation and the Neanderthals dying off, which also kind of goes with adaptation, which is our next point, that they couldn't adapt to this new climate, and it ultimately led to their starvation. And then the last thing we have is competition. So with Homo sapiens, they tended to be more innovative. They had tools like sewing kits. They were able to make clothes for the harsh environments. They also had tools like bow and arrows, which could help them hunt food and gather resources. Okay, so for our next topic on current research, we're going to be talking about speech and hearing and how researchers want to determine if Neanderthals could actually hear and speak as the same as modern day humans. So the first thing they did was they did high resolution CT scans, and they used that in order to create 3D models of Neanderthal ear structures in order to determine the optimal hearing frequencies that they could hear. And it's important to note that human speech occurs around 5 kilohertz frequency, and using these 3D structures, they were able to determine that Neanderthals had a greater sensitivity to frequencies around 4 to 5 kilohertz, which basically concludes that Neanderthals had the same hearing capabilities as modern humans today. Okay, so because researchers actually concluded that Neanderthals have the capacity to hear the same as humans, this suggests that they could potentially speak as well. And this kind of raises the question, did they have their own language? Which it's hard to determine, but there is strong evidence, since they were able to hear and speak the same as humans, that they could potentially have developed their own language and different forms of communication as well. Okay, so now we can actually look at some insights of research, and these, what we have listed, are actually kind of difficulties of researching Neanderthal DNA. The first one we have is the rarity of actually finding good samples for DNA extraction. Obviously, bones are very scarce and it's very hard to find, and it's hard to find a good, perfect sample. Then the next thing would be that we would have a low DNA yield because of contamination. It's also important to note that these bones have been in the ground for 400,000 years, and they have to be almost perfectly preserved in order to get a good sample out of them. And both of these things kind of lead into the actual frequency of the research. Not much research is actually able to be done because it's so hard to find good quality samples. And because of that, we want to make sure that the actual quality of research is high and that we take precautions and are very careful and do the right things.